Hello everyone, welcome back to my channel. So this is part 4 video of quick and concise notes of plant physiology chapter photosynthesis in higher plants. I have already uploaded part 1, part 2 and part 3 video. So please go and watch them first for better understanding. So let's start this part. So the topic is Calvin cycle. Calvin cycle basically occurs in all the photosynthetic plants, be it C3 and C4 both. There are basically 3 steps of Calvin cycle. First is carboxylation, then reduction, and then regeneration. First, let's talk about carboxylation. Carboxylation is the most crucial step of your Calvin cycle. Here, carbon dioxide is utilized for the carboxylation of RUBP, ribulose bisphosphate carboxylase enzyme, and this results in the formation of 2 molecules of 3-phosphoglyceric acid. The main important enzyme here is Rubisco, which is your RUBP carboxylase oxygenase. Next is reduction. Reduction is the second step of Calvin cycle. Here, a series of reactions takes place which leads to the formation of glucose. 2 ATP are utilized for phosphorylation reaction and 2 NADPH are used for the reduction per CO2 molecules. Here, for the fixation of 6 carbon dioxide, 6 turns of the cycle are required, which leads to removal of 1 glucose molecules. You will better understand this when we will start the formulas of this Calvin cycle. Now the third step of Calvin cycle is regeneration. Here the carbon dioxide acceptor molecule is RUBP, which is the most crucial, and 1 ATP is utilized here also for phosphorylation to form RUBP. This is the byproduct. Now let's summarize all the formulas together. 1 carbon dioxide leads to the formation of 3 ATP and 2 NADPH. Similarly, for 6 carbon dioxide, 18 ATP and 12 NADPH will be required. For 1 glucose molecule, we require 18 ADP and 12 NADP reduction. And for 1 glucose, again 6 turns of the cycle are required. So these formulas are very important. Numerical questions usually comes from these formulas. Next is C4 plants. The C4 plants are found in dry regions and they have a special leaf anatomy which is called Kranz anatomy. Here the common features of C4 plants are that they can tolerate higher temperature, high light intensities, and they also lack photorespiration. They have a greater productivity of biomass. The bundle sheath cells of C4 plants consist of large cells around the vascular bundle part. They also have large number of chloroplasts and thick walls without any intercellular spaces. Next important points about C4 plants are that the primary carbon dioxide acceptor of C4 plants is a 3-carbon phosphoenol pyruvate. This is a carboxylase enzyme. So, byproduct that we are going to get is oxaloacetic acid, which is formed in the mesophyll cells. 2 types of acids, C4 acids, are malic acid and aspartic acid. These are also formed in the mesophyll cells. The C4 acids are transported to the bundle sheath cells, which is present in the C4 plants, and they further are broken down into carbon dioxide and C3 acids. The C3 acids again go to the mesophyll cells, which again goes to the PEP, that is the primary CO2 acceptor, and so this way the cycle continues. The bundle sheath cells also are rich in Rubisco enzyme and they lack pepcase. This is very important point. Next important topic is the last topic of this chapter, which is the Blackman's law. This topic mainly includes all the factors which are responsible to affect the rate of photosynthesis. So first factor is light. All know the normal photosynthetic active radiation values, which is 400 nanometer to 700 nanometer. So if a light of higher intensity is there, it leads to breakdown of chlorophyll, which will decrease the photosynthesis. This is only exception case here is shady areas where light is rarely a limiting factor. Second important factor is carbon dioxide. This is the major limiting factor. Increase in concentration of carbon dioxide up to 0.05% also will lead to increase in carbon dioxide fixation rate, and if it is beyond this point, then that leads to damage of the plants. In low light conditions, both C3 and C4 plants will not respond, so this will lead to higher CO2 conditions. Higher lights, both C3 and C4 plants will slow down and increases in the photosynthetic rate. Third factor is temperature. In dark reaction, usually the temperature is in a controlled condition because it is an enzymatic reaction. The C4 plants usually respond to increase in temperature, which leads to increase in rate of photosynthesis. C3 plants usually have low temperature optimums. Fourth factor is water. Water stress leads to stomata to close down. On leaves, there will be wilting, and the leaf surface area will also decrease, and the metabolic activity of the plant will also decrease. So these three factors are very important. These leads to the water stress and affect the plant. So that marks the end of this chapter. I hope you all understood it. Thanks for watching.